Hello, everyone. Welcome to today's live broadcast, Precision Genomics and Precision Medicine for Immuno-Oncology. My name is Toby Ward, and I'm a field application scientist at Personalis Incorporated, and I'll be your moderator for today's event. We are pleased to bring you this educational web seminar presented by LabRoots and sponsored by Personalis, a full-solution genomic sequencing company in Menlo Park, California. For more information, we invite you to please visit personalis.com. We do have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit your questions by typing them into the Q&A box, which is located in the lower left-hand of your presentation window, and there's a green Q&A button there. I will cue the questions as they come in, and we will try to answer as many as we can at the end of the presentation. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window, or submit your problem through the green Q&A button at the lower left. This is an educational webinar and thus offers free continuing medical education credits such as PACE, RACE, CME, or CEU. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your webpage and follow the process to obtain your credits. I'd now like to introduce today's speaker, Dr. Erin Newburn. Erin joined Personalis as a field application scientist in 2013 with over 12 years of research experience in areas of molecular biology, genetics, and biotechnology. Erin's field applications team at Personalis has the responsibility for providing both pre- and post-sales technical support for the ACE cancer portfolio. She completed her postdoctoral training at the National Institute of Mental Health, where she investigated candidate susceptibility genes for major psychiatric illnesses. And she received her PhD from The Ohio State University in Integrated Biomedical Sciences as a presidential fellow. Today, Erin will discuss precision genomics and precision medicine for immuno-oncology. The learning objectives for the presentation are first, to review opportunities for precision medicine and immuno-oncology from checkpoint modulators to adoptive cell therapy. Second, to understand the key accuracy challenges in utilizing technologies like next-generation sequencing for developing particular immunotherapies, such as personalized cancer vaccines. And finally, to evaluate the current methods to predict likelihood of a positive response to immunotherapies. And now I'd like to turn it over to Erin for her presentation.
Thanks, Toby, for that introduction, and thanks to the audience for joining us today. So before we get going, I'd like to just reiterate the learning objectives that Toby just mentioned that we'll be touching on during the presentation. So we'll be talking about opportunities for precision medicine in immuno-oncology. We'll be discussing some of the current methods out there to predict response to immunotherapies. So here today I'm going to be focusing on PD-L1 status, tumor mutational burden, and also talk about neoantigen burden as an example of current predictors. And then I'll be highlighting some of the challenges in utilizing technology, such as next-generation sequencing, for the development within the immuno-oncology space. So this is a tremendously exciting time for precision medicine and for therapy in cancer. It really is the moonshot of our times. And so the National Cancer Moonshot Initiative is looking to make more therapies available for patients, but also to really improve upon our ability to prevent cancer and also to detect it early. But precision medicine is already happening in cancer in specific areas of treatment. So an example is in targeted therapy diagnostics. So Personalis and other companies are sequencing tumors, identifying the somatic mutations within the tumors, and then using that information to guide the choice of specific targeted therapies. And this is very beneficial for both patients and physicians. And these types of tests are tests that can be ordered today. But there is a whole other area of cancer treatment that is really proving to be very transformational, and that is immuno-oncology and immunotherapies that actually harness and augment our own immune system's ability to fight cancer. So you may have heard that former President Jimmy Carter was diagnosed with metastatic melanoma that had spread to his brain and liver. And unfortunately, he was given just several months to live last year. However, he received one of these immunotherapies and now is considered to be disease-free. And this has been for a type of cancer that really in the past has defied really meaningful treatment. And so it's not surprising that the area of immunotherapy is really exploding. And on this slide, I've categorized immunotherapies into three main areas for our presentation. So the first is immune checkpoints and immune modulators. So currently there are a variety of immune checkpoints being investigated, such as PD-L1, CTLA-4, and there are other very promising candidates out there as well. There's also a next generation of cancer vaccines, as well as adoptive cell therapies that have been very effective. However, the fact remains that there are still individuals that respond to these immunotherapy treatments, and unfortunately, some that do not. And so as these therapies really continue to evolve and expand, it becomes really important, increasingly important, to know which of these therapies are going to work for a particular tumor type and also going to work for an individual patient. So determining which patients will derive benefit from immunotherapy is an extremely important clinical question to really try to spare patients from ineffective treatment, but also to limit their exposure to these treatment adverse effects. So Personalis is starting to determine how we can develop precision medicine solutions to guide immunotherapy. So for our talk today, I'd like to focus on two particular examples. So the first example, immune checkpoints and immune modulators. And then I'll also be talking and discussing with you personalized neoantigen cancer vaccines. So first, let's discuss immune checkpoint blockade. So tumors are eliminated by our immune system by active surveillance, but eventually they can actually escape our immune system. And we really didn't know the molecular mechanisms behind this until more recently. So our immune system is able to recognize and is ready to eliminate cancer, but it can be held back by these inhibitory receptors and ligands. So tumors can begin to express these immune checkpoint genes, and then this creates a way then for the tumor to evade the immune system. So this is an active area of new drug development. So drugs that interrupt these immune checkpoints, such as anti-CTLA-4 or anti-PD-L1, can unleash anti-tumor immunity and really mediate cancer regression. So shown here on the screen, this is a timeline taken from a recent review. And as you can see here in this timeline, published in Nature Reviews, there have been very exciting milestones that have been reached in the generation of these checkpoint drugs. And the responses can be very dramatic and lead to lasting remission in cancers that, at least traditionally, have been difficult to treat. However, the objective response rate still ranges from approximately 10% to 30%. So the question again is, how do we really predict who is going to respond to these immune checkpoint therapies? So the first generation of attempts in predictive biomarkers for immunotherapies have resulted in the single gene expression test. So looking at the expression of a single marker, so here PD-L1 status is being determined. And in this case, you can see that if the test is positive, you have a 26% chance of responding to this checkpoint inhibitor drug. But what you can also see is that if you're negative, you have a 10% chance of responding. So patients negative for this biomarker may still respond. And so this really complicates the issue of using PD-L1 as an exclusive biomarker, exclusive predictor. And so, these types of tests are coarse predictors at best and relatively underwhelming. So the question becomes, how do we build better predictors? And this is what Personalis and others are working on. The modulators of the immune system and immune response are extremely complex and go well beyond a single gene. And so this raises the opportunity then to use more sophisticated, broader genomics as well as informatic methods to really drive this next generation of precision medicine solutions. And so there are currently other ideas out there about better signatures, better predictors to determine the likelihood of response to these immunotherapies. So there have been studies showing that perhaps mutational burden as a signature that can potentially predict response to these immunotherapies. So shown here on the screen is a snapshot from a New England Journal of Medicine article using ipilimumab for patients with melanoma. And here, this is the Kaplan-Meier survival curve showing response of patients on this anti-CTLA-4 therapy correlated with a higher mutational burden. And you can see that there on the blue line, as compared to individuals with a lower number of mutations shown in red. And so there have also been studies showing that perhaps neoantigen burden may also be used as a potential predictor of response to immunotherapies. And while tumor mutational burden is the total number of mutations in the coding region of the tumor genome, a subset of these mutations may result in neoantigens. So neoantigens, these are somatic mutations in cancer that can generate non-self peptides, and when properly presented on HLA complex, can be targets for tumor-specific T-cell responses. So as an example, shown here is a study published on patients with non-small cell lung cancer receiving anti-PD-1 therapy. And they show a higher percent of progression-free survival in tumors with higher candidate neoantigen burden as compared to tumors with a lower candidate neoantigen burden. So could neoantigen burden perhaps then be a more refined predictor for determining response to these therapies? Well, there seems to be limited data so far in the literature, but perhaps that is because the neoantigen universe has yet to be clearly defined. So making sure we include both classes of neoantigens, both class 1 and class 2, as well as making sure that the assays that we use in this assessment are accurate in detection, and then also that all variant types are considered in this assessment. So as we search for better molecular determinants of response to immunotherapies, what genomic assay or footprint do you need to determine mutational load or to determine neoantigen load? So for mutational load determination, this year at the 2016 ASCO meeting, the ability to extrapolate total mutational burden using a large panel was really highlighted. And there have been several papers and studies recently to support this idea. A more recent question out there is, what footprint do you need for a neoantigen load assessment? And this was a very interesting question that was raised from this recent study in genome medicine. So using samples from patients with lung or colon adenocarcinoma for mutational burden assessment, they demonstrated that a large panel, and here this was a panel of 300 genes, was highly correlated with the whole exome sequencing results for mutational burden assessment. However, when looking at the ability then to predict neoantigen load from those same samples, using whole exome sequencing as compared to this large panel, neoantigen predictions from this large panel were very limited. Most diagnostic panels consist of cancer driver genes, and many neoantigens discovered are outside of the typical cancer driver genes. So how do you get at these neoantigens? How do you assess these? So as demonstrated from the study on the previous slide, the existing panels are really too limited for neoantigen detection. These existing assays are really narrow, as they typically contain 20 or maybe up to 500 genes. But also, oftentimes with detecting these neoantigens, the technology is limited by incomplete coverage of genes. So that could lead to missed potential mutations, and then this can lead to limitations in accurate predictions of neoantigens. So at Personalis, we've developed a platform for accurately detecting neoantigens. So our ImmunoID is an example of a platform that is broader, more sophisticated, and can really help power this next generation of precision medicine in immuno-oncology. So we've been very innovative in a number of different areas. The first is that we created and patented the ACE technology to achieve clinical grade sequencing, and I'll be talking more about that in the presentation. We've also went beyond the model of looking at one gene or a few genes at a time, but really try to broaden out and interrogate all 20,000 plus genes. And this also involves integration of both DNA and RNA together, and that feeds into the algorithms that we put together for neoantigen detection. And at the point here really is that the ImmunoID platform allows the beginning of the determination of metrics like neoantigen burden or other biomarkers that could eventually correlate with immunotherapy response and serve as this next generation of precision diagnostics. So developing the technology in precision medicine is really just the start. So validation and regulatory issues are key if you're going to really use these tests at the bedside. So strong analytical validation is required, but this is oftentimes challenging in cancer because there's a lack of existing gold standards or metrics to really assess performance. So our feelings as a company at Personalis are that we need to move away from this wild west of genomics and spend really time doing a very robust analytical validation, and this is very critical. So some of the Personalis scientific founders co-authored this article pictured here on the screen with individuals from the FDA that really laid out a roadmap of regulatory or considerations for how to regulate within this space. So at Personalis, we've recognized the need for clinical grade sequencing and informatics as well to support diagnostic use. And so in the next set of slides, I'd like to speak to how we've addressed this need. So first, let's look at sequencing assays. So one of the biggest challenges in standard whole exome sequencing assays are the gaps and inconsistencies in the coverage. So for example, this is a sequencing plot shown here on your screen of the STK11 gene using standard exome sequencing. So this is the Agilent Clinical Research Exome Assay that was used. And the plot shows all the exons concatenated together, and the read depth is shown here on your y-axis. And so as you notice, although this gene is sequenced at a high depth, and so in this case we're looking at an average of greater than 100x coverage, it still fails to cover whole exonic regions, and you can see that there in the red brackets as shown. And so variants that reside in these regions would be completely missed. So we've recognized these issues with the standard exome and built the ACE exome design that works by taking the standard exome, shown here in blue on the schematic, and adding supplemental coverage in green for those problematic areas in the genome that are difficult to cover. So these are combined then in an effort to maximize genomic content and really provide uniformity in coverage to increase confidence in calling variants. Also, not only do we target exonic content, but we've also augmented coverage of biomedically important regions outside the exon, such as regulatory regions, intergenic, or intronic areas. So if I go back to STK11, this is a nice visual of the quality of data that you would receive from an enhanced exome approach such as ours. So we provide you with the blue plus the green supplemental coverage, and this results in more accurate, more uniform coverage across this critical gene. And so, and with our ACE augmentation, we go beyond the cancer driver genes and finish over 8,000 biomedically important genes where random somatic mutations can occur. And here are some more sequencing plots just to show you a visual of a variety of genes with gaps, inconsistencies in coverage that are very nicely filled in by the ACE technology. And this is particularly important for immuno-oncology studies. And our augmented exome approach that we use was recently highlighted in Nature Reviews Genetics in an article titled Towards Precision Medicine. So the author mentions the challenges with standard exome assays as coverage of certain areas can be very limited, like GC-rich regions. And they show a gene here that is poorly covered with a standard exome assay. So this is a critical gene, KCNH2, a potassium channel gene that is associated with long and short QT syndrome. And with an augmented approach such as ours, we have more uniformity across this critical gene than the standard assays. Likewise, accurate identification of the expressed somatic variants using whole exome sequencing is critical as you are defining your genomic solutions for precision medicine. In many of these cancer studies, formalin-fixed, paraffin-embedded samples are the materials available for analysis. And at least in the past, these sample types have been quite challenging for genomic and most definitely for transcriptomic studies. So typical enrichment methods such as poly-A ribodepletion have limitations, such as inconsistent removal of these ribosomal RNA reads, or just the inability to work with these more degraded sample types. So methods such as our exome capture transcriptome protocol, the ACE cancer transcriptome, allows you to overcome these limitations. So again, RNA-seq is quite critical for neoantigen detection, as you really want to determine if these variants are indeed expressed variants. So using an exome capture transcriptome protocol, and here are 4 different cancer genes shown on the screen, this enables more sensitive variant detection. So in blue, you can see the transcriptome coverage using a standard RNA-seq methodology, whereas using an optimized exome capture transcriptome protocol, the blue plus the green, provides more sequencing coverage across these critical cancer genes for increased sensitivity for somatic variant detection. So next, I'd also like to further highlight that strong analytical validation of the assays and the informatics for these precision medicine solutions is critical. I want to share just a few slides on the approach we use for analytical validation of our enhanced exome for use in neoantigen detection. So in these next set of slides, I'll be focusing on our approach for our ACE cancer exome assay, but we also have completed a validation of our transcriptome as well. So as an example of this process, I'm going to walk through how we assessed analytical sensitivity for small variants. So the positive percent agreement and the positive predictive value were computed over a range of minor allele frequencies. And one of the major challenges facing the industry when you're performing these types of analytical validations is the lack of an established gold standard or reference samples that can be used as a ground truth. So we developed a validation approach where we used well-characterized cancer cell lines to generate these reference samples that could represent variant types. And so in our study, we used a set of gold standards, and we refer to our gold set as a set of cosmic verified events. So this means that they were detected in the Cosmic Cell Line Project and also verified by 2 other sources. So Cancer Cell Line Encyclopedia, the NCI 60, or then confirmed by Sanger sequencing. So in total here, across the 11 cell lines that we used for our exome validation, the gold set that we defined here looked at 875 SNVs and 19 indels. And so in our approach here, the 11 specific cancer cell lines also had the corresponding matched normal cell lines available. So we sequenced these 11 pairs, then ran our somatic tumor normal pipeline, and then we compared these variant results to the gold set of standards. So we also looked at our limits of detection of these small variants using 3 pairs of tumor and normal cell lines. We created dilutions to represent a 5% up to an 80% tumor purity, as you see here on the screen. So for example, we took 5% of the tumor cell line, mixed that with 95% of the normal cell line to simulate a 5% tumor purity, and so on here up to an 80% tumor purity. And so I've shared here just a brief overview of our approach for our exome. And as I mentioned, we've completed the analytical validation of our transcriptome as well. And we feel that it's critical, very important to measure the performance of these assays that are being used. And so from that plan, we are very pleased to share these final results for the small variants. So our overall analytical sensitivity for SNVs and indels using our ACE cancer exome is 98% and 95% respectively. And then what we've done here on the screen in the second table is show the PPA and the PPV, and it's specifically parsed out here for the SNVs and the indel results, as well as the value for the overall for small variants. So you can see this at a 10% minor allele frequency, as well as at a 20% minor allele frequency. And as you can see here, our results are quite good. So now that you have accurate clinical grade comprehensive sequencing data, how then do you identify and predict potential neoantigens? So this is a workflow here for our process to enable accurate candidate neoantigen detection. So it starts here by taking a cancer sample and a matched normal sample from the patient. We sequence that with our enhanced ACE cancer exome that I just described to you. And this sequencing data is then analyzed through our somatic analysis pipeline to generate the set of all somatic variants. We then use RNA derived from those tumor samples to perform our enhanced whole transcriptome sequencing. So somatic variants are then called in the RNA as well, and then can be cross-referenced with the somatic variants called in the DNA. And this then allows the ability to look at the set of all somatic variants that are actually expressed and therefore could be potential neoantigen candidates. And we believe that the assays that are being used in this process can really make a difference. And here's an example of a candidate neoantigen identified in a patient sample. So you can see the coverage above here of this blue-green plot of the DNA and RNA coverage for this variant. So this was a missense mutation in the RPS2 gene. And the red line there shows you exactly where this candidate neoantigen resides. And it's quite possible that this variant would have been undetected by a standard assay. But with an augmented exome approach such as ours, it provides greater sensitivity to detect these candidate neoantigens. And so one of the very exciting pieces in this space is the ability to further predict neoantigens through deeper bioinformatic analysis. So at Personalis, we're currently developing a neoantigen prediction pipeline. And for us, it starts by imputing into this algorithm, our pipeline, our high quality data. So utilizing our optimized exome to sequence the tumor in the matched normal, we perform epitope prediction. Then we also perform in silico HLA typing to determine MHC binding capabilities. And then in parallel, using our optimized ACE transcriptome, we use the gene expression, the variant expression information to determine similarity to known antigens as well as similarity to self. So this then, all these pieces then will be used to rank and report out candidate neoantigens. And so far we've spoken about neoantigens in the context of precision medicine for checkpoint blockade therapies. So this is the idea of using neoantigen burden as a predictive signature. However, there is another entire application of neoantigens, which is in creating truly personalized cancer vaccines. So for personalized cancer vaccine development, you can identify the neoantigen and then create a synthetic vaccine that is personalized for this particular tumor, and then provide that to the patient. And oftentimes this vaccine is provided in combination with these checkpoint blockade therapies, as well as can be provided with targeted therapies or chemotherapies as well. And so here you have a tumor-specific personalized cancer vaccine created for this individual patient. So in summary, as we conclude our presentation and open it up for some Q&A, Personalis and other companies are already working on precision diagnostics for cancer and targeted therapies. But there is a new area, immunotherapy, that is incredibly exciting and transformational, and there are tremendous opportunities for precision medicine in that realm as well. And so as a company, we're working on solutions to help predict who will respond to checkpoint blockade therapies, and also working on solutions that will drive the development of personalized cancer vaccines. And as we look to the future, it starts by making sure that the assays as well as the informatics involved in this process are of the highest quality and highest accuracy. So if you'd like to learn more about the solutions we've designed within our portfolio, please visit our virtual booth at the LabRoots conference today. And you can also message us at info@personalis.com. Thanks.
Erin, thank you for that informative presentation. Before we get started on the Q&A session, I'd like to remind our audience how to submit their questions. You can submit your questions by typing them into the Q&A box, which is found at the lower left hand of your presentation window at the green Q&A button. We'll try to answer as many questions as we can. Our first question that we already received is, do you require a matched normal sample for your processes? And if so, what types of tissues do you accept as a matched normal?
Yeah, so in the neoantigen pipeline that I showed you, you saw that we do take both the tumor as well as the matched normal in this process, and this really increases the specificity of the somatic analysis. However, depending on your project, depending on your application, we can work with you on that. Our bioinformatics capabilities are very versatile. We also have a pipeline that's configured to work when you don't have that matched normal available. So in that case, we use a proxy normal and a database, as well as some bioinformatic filters to help compensate when, unfortunately, that matched normal might not be available.
Great. Thank you.
Oh, and I think you also asked the types of...
what types of tissues do you accept?
Yeah, so we accept blood as the matched normal. We can also take normal adjacent tissue as well.
Great, thank you. The second question that we got was, what are the typical deliverables that you provide back after a sequencing project?
Yeah, and again, we can work with you and talk with you specifically about what you may require, but we do have very comprehensive deliverables. We give you, the raw data, the FASTQ files, the alignment files, the variant call files, as well as the full somatic pipeline. We have a very nice sequencing QC report that we can deliver. And then also for RNA, we do provide gene expression values as well as gene fusion information. So we have a list of standard deliverables. However, we can also work and tailor and customize output depending on what your needs might be. So I'd encourage folks to visit our booth today or to message us, and we can talk to you about some specific output requirements and needs that you might have.
Great. A related question is, what are the standard turnaround times for the assays that you've offered today?
Okay. Yeah, and again, this can oftentimes, it depends on the project and the application at hand. We do, for clinical trials, can offer a very expedited turnaround time of 14 calendar days if needed. For more standard products, such as if it's a research study, we typically have about a 4 to 6 week turnaround time. But again, these can be based on your needs as well.
Okay, here's a technical question. For your immuno-oncology applications, do you have a recommended depth of sequencing that you think is sufficient to drive these projects?
Yeah, so in the validation that I presented here, the approach we used for the validation, this validation was specifically geared for neoantigen detection. And in this case, we are typically sequencing the tumor to a 200x type coverage and pairing that with about 100x or so of the matched normal sample. And so for certain applications, of course, a higher depth like vaccine development may be required, but we also offer a variety of different sequencing outputs and depths, depending, again, upon the study need.
Okay, we have time for one last question. And that question is, do you offer other assays besides this whole exome and whole transcriptome configuration?
Yeah, so there wasn't time today, but we do. So we offer our enhanced exome and transcriptome, which gives you As you can see, very broad profiling, and we've optimized for accuracy within that assay, and some obviously definite applications for immuno-oncology. However, we also have an enhanced cancer panel for both DNA and RNA. It's currently the most comprehensive cancer panel out there on the market. It's 1,400 cancer genes, and we do both DNA and RNA of that panel. Within the design of that panel, we've really tried to make sure that we include, very critical genes, and as well as include very critical cancer pathway analysis. And so this panel has definitely suited for targeted therapy applications as well as immuno-oncology applications. So today we were talking about the ability of a panel to extrapolate mutational burden assessment, and this would be possible with our panel.
All right. I'd like to once again thank Dr. Newburn for her presentation. Would you like to make any final comments?
Thanks for everybody for joining us today, and please check out our booth today at the virtual conference.
Okay. I would also like to thank our sponsor, Personalis Incorporated, for making today's educational webcast possible. Please visit personalis.com for more information about our company and our capabilities. As a reminder, today's webcast will be available for on-demand viewing for 6 months from the live date until April 2017. You should receive an email from LabRoots alerting you when this webcast will be available for replay, and we do invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Goodbye.